Hey everyone, welcome back to another episode of Genetics. Today we're going to go over analyzing PCR, variable number tandem repeats, how to analyze the jellies that we get from that, and then how do we analyze a Sanger sequence. To begin with, we're going to start off with the VNTRs. So typically the way what they're doing over here, is essentially they're taking every allele has essentially a different amount of tandem repeats, and what you can do with these tandem repeats is that every single one of these alleles are all going to be a different molecular weight, and because they're all a different molecular weight, they're going to appear differently on a gel electrophoresis. So what you can do with these is that you can analyze the alleles from one individual, and you can use that to determine where that allele came from other related individuals. So hopefully that kind of made sense. So basically, what this method is used for is mostly in forensics, and and in these forensics, you're using this to analyze which individual committed a certain crime based on the number of repeats. Okay, so to analyze this kind of a problem, what do we have here? And the questions typically go up to is, who's the parents of this offspring right here? And the way how you would typically answer this question is you would look at the alleles from this individual, from the offspring, and you're going to look at their weight. And these questions are pretty simple, to be honest, but a lot of people struggle on it because if they overcomplicate it. And what you want to do is you want to look at where does these alleles come from and from what parent, right? So if you look at the top one over here, that allele most likely came from either male 2 or female 2, right? But we don't know which one it is. So what you have to do now is you have to look at the other allele from this offspring. And this other allele from this offspring came from male 1, and that's the only possibility, right? So because the bottom allele came from male 1, therefore the top allele has to have come from a female. So therefore the top allele had to have come from female 2. And that's pretty much the gist of how complicated these kinds of problems would be for an exam. They wouldn't be anything too complicated. But essentially, this is how you're going to answer these questions. So we would say male 1 and female 2 are parents of the offspring. So that's basically how you do it. Pretty simple, pretty self-explanatory, but some people do struggle on that as well. Next up, this is where all goes loose, okay? In an exam question, what I've seen from professors is that you would get something like this. You would get a sequence, and we can say that the box over here is basically the gene of interest. And the question is, what would happen in the next round of polymerase chain reaction? So remember, polymerase chain reaction is basically it's basically just replication, right? DNA replication. We've covered DNA replication in a previous episode on this channel. So to answer this question, we basically have to understand 3 different things. The first thing that you want to understand is that primers go on genes. The second thing to understand from this type of a question is that DNA polymerase or Taq polymerase, doesn't really matter, reads DNA from 3 to 5, and it builds new strand 5 to 3. Right? And then the last thing is that every single one of these primers, they're always going to end up on the 3' end. So primers always go on 3' end. Okay? So basically, to answer, we're going to look at the one on the left. And what we can say here, that the squiggly line, this section right here, is basically just random DNA. And what we're trying to amplify is the box segment, okay? So the box is the gene. So we can draw it out over here again. So 5' squiggly line. So like I said, first of all, your primers are always going to go on the gene that you want to amplify, okay? And additionally, those primers will always end up on the 3' end of that gene, okay? So basically, during our replication cycle, what will happen here is that the primer will basically get attached to this 3' end of the gene, and then if we look at rule number 2, which states that DNA polymerase reads DNA from 3 to 5, basically DNA polymerase 3 will read the DNA sequence going this way, right? And this new DNA sequence is basically going to be 5 to 3. Basically, from the point that you've added the primer, that'll be your 5' end. And then when you've ran out of DNA, that'll be your 3' end, okay? So basically, to answer this question, especially the one on the left, you would always have 2 different answers for these questions. Basically, one answer will have, because we're looking at the PCR product, one of the answers will be the exact same sequence. The reason why it will be the exact same sequence is because we're following the semi-conservative model of replication. So therefore, we would expect to have a newly replicated strand and the original strand as well, always in your answer. So with these questions, you should always have the original strand written down on your exam. Otherwise, you're just missing out on easy points. Okay. Now next, what you have to do is essentially you just have to copy and paste what you have written over here. So basically, because we've started our primer at the gene that we're trying to amplify, and we haven't amplified the unnecessary region over here, therefore we don't have to write that down. But because we've copied this section over here, because these enzymes don't actually know where the end of these DNA sequence, or where the end of your gene is. So what they're going to do is they're going to continuously replicate until they've run out of space to replicate. So basically they're going to continuously replicate to get us this answer for our problem. So hopefully that kind of makes sense. We can do another problem over here. I'm going to write it down real quick. So for this problem right here, again, what we're going to do is we're going to add our primer at the 3' end, which is over here. And then we're always going to read DNA polymerase. DNA polymerase is always going to read 3 to 5. So it's going to go this way. And then this new sequence will be 5 to 3. So our answer would be, for this question, will be the original strand, 3 to 5, and then our newly replicated strand, which we've written down over here. So 5 3 prime. That's it. So hopefully that made sense. And typically this is what I see most of the time. You may be given a different format for this kind of a question, but basically, if you understand these 3 different rules, you can answer any PCR related question on your exam. Okay. And I always recommend if you don't really understand the rules, try your best and provide yourself with different kinds of questions that you can have. There are some good questions on your textbook that you can try as well. So I really recommend working your best. Try to figure out a way how you can succeed with this PCR related question. Okay. And then next up, Sanger sequencing. Now this one is a weird one because sometimes some professors have different requirements for this kind of a question, and some don't. Most of the time, what I've seen is that depending on what your professor wants you to do, some professors just give you the sequence of a particular gene, and then just analyze what the sequence is from 5 to 3. Some don't. You have to do a little bit more work, and we're going to go over that real quick. But essentially, how you read these Sanger sequences, and if you're not really aware of these, basically a Sanger sequence is a methodology that you can use to figure out the exact DNA sequence of a particular gene. That's basically all it is. And then you put it like on this gel with different lanes, and we're not going to go into the specifics of Sanger sequencing, but essentially this is how your Sanger sequencing, or how your Sanger sequence will look on the gel. And remember how in the previous problem, DNA polymerase will always begin making the newly synthesized strand of DNA from 5 to 3. So, and the way how this gel works is that typically the bottom of the gel is written down as 5 prime, and then the top part of the gel is written down as 3 prime. So the reason why this is the case is because remember how DNA polymerase makes a new strand of DNA going to 5 to 3, and what's happening here is, like, let's imagine we have our sequence that we have up top here, and then we start making a newly synthesized strand with DNA polymerase. So we add our primer, and then this newly synthesized strand will always be 5 to 3, and then let's say we add a T here. And then you would randomly stop that reaction at that point in time, and you would put that onto a gel, and then that will go all the way to the bottom because it has the least molecular weight, because it only has one nucleotide there. Okay? We don't have to go into the specifics of it because, frankly, not a lot of people ask specific questions on these particular methodologies. They just ask you to analyze stuff, okay? So basically, looking at that, what you can do here is that your sequence is always going to be 5 to 3, and what you have to do here is basically just read it 5 to 3. So you would always begin 5 prime, T, C, and let me keep track so you can see as well. Or let me just write it down in red, why not? 5 prime, T, C, A, G, T, G, T, 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 G, T, C, T, C, G, G, T, A, C, A, T. So then don't forget your last 3' end right there. And this is basically how you read a Sanger sequencing. How you read a Sanger sequence. It's pretty simple. All right, so this is just a general way how you read a Sanger sequencing. So here comes the part where it might be a little difficult for you to sort of grasp on what an exam question is asking you. So an exam question, sometimes what I've seen is that they might ask you, is this sequence that we've sequenced right here, is that the template or is that the coding strand of the gene? So is this the template or the coding strand of gene? So how do we answer that question? So here's the thing that you have to know is that the coding strand always has ATG, or it always has a start codon and a stop codon, unless otherwise specified. So some professors will tell you beforehand that a sequence doesn't have a start or a stop. So just be aware of that. Make sure you read the question carefully. So what we need to do is we need to figure out if this is the coding sequence or the template sequence, okay? So so the way how you begin this is by looking at the start codon. The start codon is always organized in a way that it's 5' AUG, okay? So, and you can see this on the codon chart as well. You always read the codon chart from the perspective of the mRNA, and you always, and it's always in the 5' to 3' orientation. So so what we're looking for is basically anything that is 5' AUG. In other words, another thing that you have to look at is the inverse of that, because remember, in these cells there really isn't a left or a right way of how to read a DNA sequence. It's only in the direction of the 5' orientation. So basically you have to be very observant and know that this is the same thing as this, okay? Remember that in these cells they don't have a left or a right. They only have the orientation given to them by the molecules, and then this orientation will be used for you to figure out where a 5' AUG is, right? So if you're reading it going, if you're reading it like the bottom one where you're going left to right, always be sure to just take it one step at a time. We would read it as 5' AUG, and you have your start codon, okay? The one on the left, or the one on the top, is pretty self-explanatory. You've done it a million times. You read it left to right. So what we're going to do is we're going to figure out if what we have over here is the coding strand, okay? So we're going to look for, because we're always going to start on the 5' end, we're going to go left to right in this situation. And you're going to look for an AUG. But notice how we have T's in this sequence over here. So remember, another thing that you should know is that the coding strand is basically, the coding mRNA is basically the same as the coding sequence of the gene. The coding sequence of the gene. But there's one big exception. Just turn T's or U's into T's, okay? So like what you can do here is you can, on your piece of paper, you can erase all these T's into U's if you want to, or you can just use your head, and you can just use your head for this, okay? And just in your head change the T's into U's, okay? So we're gonna do that. and you're gonna try to find where you can see an ATG or an AUG, and I don't see a single one of them. So therefore, this strand that we've sequenced over here is not the coding sequence, it's the template sequence. And to double check our work, we can do that. We're going to do the complementary to the sequence. Take some time to do this real quick. So, to figure this out, to confirm our work, what we're going to do is we're going to start at the 5' end of the complementary sequence. So we're going to start, we're going to start here, and we're going to go left to right, okay? So we have an ATG, boom, that's the start codon. And then as soon as you find a start codon, we're going to go 3 nucleotides apart. So this will be our first amino acid, or our second amino acid actually, our third, our fourth, our fifth, our sixth, and our possibly 8. If you don't have your codon chart with you, this TGA is basically a UGA, and that UGA is a stop codon. So that'll be considered a stop codon. And then that's it. You figured out that the original sequence is in fact a template sequence. So good job. That's it. Hopefully this helped you out.